My abstract is part of a long story where in our group we described that almost 40% of CLL cases express the adhesion molecule named VLA-4, the very late in adhesion, number 4. And this molecule, the CLL expressing this molecule has a bad prognosis in terms of time to first treatment, overall survival, and progression-free survival. This adhesion molecule helps CLL cells to interact with the microenvironment and give a pro-survival signal to the CLL cells in the microenvironment in the tissue site. And we years ago studied the interaction of this integrin, of this adhesion molecule with the BCR, that is another player of the microenvironment. And here at that time we demonstrated that the novel therapy, the ibrutinib treatment, impair this interaction, and so the expression of this molecule go to impairing the so-called lymphocytosis recirculation that is due to the therapy, so due to the ibrutinib therapy in particular. And here in this abstract we demonstrate that this mechanism of interaction between these two molecules that is called inside-out signaling occurs also in the peripheral blood, not only in the tissue site. And so the ibrutinib, that is one of the major therapy, impairs also this interaction also in the peripheral blood, and so inhibit the capacity of the integrin to interact with its ligand that are present in this plasma, that are present in soluble way, that are become like fibronectin, and not receive anymore that proliferating signaling to that are important for the CLL cell to have it, and so to return it in the tissue site, and so in a sort of migration of the CLL cells from a tissue from another, so bone marrow lymph node. And this is a new mechanism that we described in this abstract.